In this video, you'll see how to use a hemocytometer to get an accurate cell count and how to convert this data into usable cell density and viability information. The hemocytometer was originally developed for counting blood cells. Today, it is used to count many other cell types as well. A hemocytometer consists of a special glass microscope slide that contains two separate counting chambers. Each chamber is etched with a carefully crafted grid with exact dimensions. There are two supports on either side of the counting chamber. When a cover glass is placed on the supports, the depth of the chamber will be exactly 0.1 millimeters. Now we know both the area and the depth of the counting chamber, and we can calculate the volume. We can also use the grid to count the number of cells in each section and calculate the cell density. Before you start, you need to dilute your cell suspension in trypan blue solution, which is used to stain any dead cells present in your sample. Make sure your sample is mixed well. Counts should be within 2 minutes of incubation with trypan blue. Be sure not to wait more than 5 minutes, as longer incubations will result in trypan blue entering live cells. A dilution factor between 2 and 10 usually works well for mammalian cells. To get accurate cell counts, it is recommended to have no fewer than 10 and no more than 50 cells over each square millimeter. Basically, you don't want to be counting 100 or 200 cells per square. You want to use appropriate dilutions to get 10 to 50 cells per square. Make sure the hemocytometer and cover slip is clean. 70% ethanol can be used for the cleaning. Carefully place the cover slip on the supports using gentle pressure and small circular motions. Here's a tip: moisten the shoulders of the hemocytometer before you fix the cover slip. Make sure your diluted cell sample is mixed well and load 10 microliters by gently resting the end of the pipette tip at the edge of the chamber or the edge of the cover slip. Do not overfill the chamber. Let the liquid be drawn out of the tip and fill the counting chamber by capillary action. With a microscope and a 10 times magnification, you will be able to see approximately 1 square per viewing window. Use phase contrast to distinguish the cells. Live cells will not be stained by trypan blue and will appear white, while any dead cells will be faint or dark blue. Now you can count all the cells that fall within a couple of the larger squares and include any cells on the right hand or bottom boundary line. Count the number of live cells and dead cells. A good suggestion could be to count the cells in squares 1, 3, 5, 7 and 9. Obviously, the more squares you count, the more accurate your cell count will be. Just remember how many squares you've counted. Did you know there's an app you can use for this? The Cell Culture app has a cell counter tool that will help you keep track of live and dead cell counts. Based on the number of viable and dead cells, you can now calculate the cell viability. To calculate the density of the viable cells, you need to determine the number of live cells in a particular volume. This volume is defined by the number of squares that you counted and the volume of one such square. We also multiply by the dilution factor in order to determine the cell density of your original cell solution prior to diluting with trypan blue solution. The hemocytometer is designed so that the volume of each of the larger squares is constant. Each square will hold a number of cells equivalent to the number of cells counted multiplied by 10 to the power 4 per milliliter. To convert, we then calculate the density of viable cells by looking at the number of live cells multiplied by 10 to the power 4 per milliliter, the number of squares counted, and the dilution factor. The hemocytometer and these equations help us determine cell density and viability. Based on this, you can also create growth curves or calculate the best window for cell passaging. You'll find more information on how to perform these basic cell culture calculations in a separate video.